Hello, hello. Okay. Good afternoon. So today is the last tutorial session of this Basics of Biology course, where mostly the week 12 was about summarizing the whole course content and then looking at general questions, whatever we have read. So today's tutorial session will also consist of all the summarizing, like summarizing all the contents, especially before the these different systems uh, started, as in the nervous system, digestive system. Before that part, we will be looking at different questions from all those parts, and then uh, accordingly kind of go through or revise the concepts related to the questions. So I will start sharing the screen. Hopefully the screen is visible. So now let's look. We will start with question answers only. So let's look at the first question. Which of the following statements about cellulose is correct? It is composed of alpha D glucose units. It contains beta 1,4 glycosidic bonds. It is easily digested by humans. It forms branched structures. and which of these combinations or a single option is correct or not. Those are the options. Now, the correct answer is B only. It contains beta 1,4 glycosidic bonds because cellulose is made up of beta D glucose, not alpha D glucose. And humans actually cannot digest cellulose because humans we lack cellulase enzyme, and cellulose is actually unbranched, forming straight chains. Okay. So if we look at the structure here, this is the straight chain linked by beta 1,4 glycosidic bond, and it is one of the major polysaccharides found in plants. It is the most abundant organic compound in the biosphere. And suppose if we talk about the statistics or the quantity, related quantity, then some 10 to the power 15 kg of cellulose is synthesized and degraded on Earth each year, and it is an unbranched polymer of glucose joined by beta 1,4 glycosidic linkages, and it forms this beta configuration actually allows for very long straight chains, and fibrils are formed by parallel straight chains that interact with one another by hydrogen bonding. Next, an organism shows the following features. Body parts are arranged around the central axis. The organism can be divided into similar halves by multiple planes, and the organism actually lacks a definite head and tail. So, which of the following organisms is most likely to have these features? Is it a frog, hydra, earthworm, or cockroach? Now, if we go through the options one by one, body parts arranged around the central axis, okay, can be divided into similar halves by multiple planes. Now, if you look at an earthworm or a cockroach or a frog, these show actually bilateral symmetry. It can only be, or like us humans, we have bilateral symmetry where our bodies can be divided into only one plane into like kind of equal left and right halves. However, if we talk about multiple planes here, then Hydra with its tentacles and stuff, it can be divided symmetrically into multiple halves. Okay, as in by multiple planes, it can be divided into two similar halves. So, and definitely it lacks a definite head and tail. So, this is basically the correct answer is Hydra. And the features given here indicate radial symmetry, where the body can be divided into similar halves along multiple planes passing through the central axis. Frog, earthworm, and cockroach, as I said earlier, show bilateral symmetry, where only one plane can divide the body into two equal halves. So if we look at it conceptually, if organisms can be, divided into different parameters or characteristics, especially like grade of organization, they can be unicellular or multicellular based on the number of cells, symmetry, which is the plane of arrangement of body parts. Radial symmetry and bilateral symmetry are the two types of symmetry. In radial symmetry, this one here, this is the here the organism is hydra as well as it was given in the question. In radial symmetry, the body parts are arranged around the central axis. If we cut through the central axis in any direction, it can be divided into similar halves, for example, hydra, jellyfish, and starfish. In bilateral symmetry, the body parts are arranged along a central axis here, okay? And if we cut through the central axis, we get two identical halves. For example, this turtle here, a frog, we, like humans, also have bilateral symmetry. Next, if we talk about the germ layers are formed during the development of an embryo. These layers give rise to different organisms as the embryo becomes an adult. Now, if an organism has two germ layers, the ectoderm and the endoderm, the organism is a diploblastic organism. And in case they have three germ layers, the ectoderm, the mesoderm, and endoderm, they are triploblastic animals. Next question. Consider the following statements. Diploblastic organisms, the organisms having two layers, have only ectoderm and endoderm. Triploblastic organisms have a mesoderm in addition to ectoderm and endoderm. All radially symmetrical animals are triploblastic. and higher level of organization is generally associated with a triple plastic condition. So which of the following combinations of options are correct? So diploblastic organisms, we looked at it just in the previous slide, that they have only ectoderm and endoderm. Triploblastic organisms have a mesoderm in addition to ectoderm and endoderm. Okay, so both these options are correct, A and B. However, radially symmetrical animals are not, like all radially symmetrical animals aren't necessarily triploblastic, because if you look at the example of Hydra, right? Hydra is a diploblastic organism with only ectoderm and endoderm, and there's no mesoderm present. Okay, and higher level of organization is generally associated with triploblastic condition. So if this is a correct statement, in case it was mentioned like always associated with triploblastic condition, then we could have been a little, skeptical. But like if it is talking about the general trend, then this is a correct statement. So options A, B, and D are correct. Okay. Next, we have coelom. Coelom refers to a fluid-filled cavity. Like we looked at the different layers, right? Germ layers. So based on the type of germ layers, as in how the, especially the how the germ layers, especially the mesodermal layer, is distributed or present inside the body. Uh, it also defines the cavity, body cavity, which is also called as coelom. This is the fluid-filled cavity inside the body. It separates the digestive tract and other organs from the body wall. A true body cavity or coelom is one that is located within the mesoderm. Uh, this one here, okay. And based on the nature of the coelom, again there can be three groups where organisms like earthworms are called coelomates or true coelomates because they have a true coelom or true cavity. Tapeworm here is an acoelomate where they are actually triploblastic. They have three layers: endoderm, this yellow one, the pink one, the mesoderm, and the blue one, the ectoderm. However, there's no division in between in the mesodermal layer or any separation in the mesodermal layer giving rise to the body cavity. It's only the digestive cavity present here. Okay. And then there are animals like roundworm, which have a body cavity, but it is located between the endoderm and mesoderm. Okay. Pseudocoelomates, what is happening here is there's mesoderm, there's endoderm, and there's a gap between endoderm and mesoderm. Okay. So in this scenario, in the case of a roundworm, it has a false coelom, and such organisms are called as pseudocoelomates. We come to the next question, which is in an experiment similar to that of Louis Pasteur, nutrient broth was boiled and placed in a swan neck flask. The flask remained open to air, but no microbial growth was observed unless the neck was broken. So, which of the following conclusions is most appropriate? Air contains no microbes. Microbes arise spontaneously from non living matter. Microbes enter the broth from air but are trapped in the curved neck. Boiling permanently destroys the ability of broth to support life. So, this is actually based on the different theories of evolution, and where uh, there were different kinds of theories. Out of them, one was the theory of spontaneous generation, which was actually kind of disproved by different people. One of them being Louis Pasteur. So this is based on his swan neck experiment. So the correct option is option 3. So Louis Pasteur designed the swan neck flask experiment to disprove spontaneous generation. The curved neck allowed air to enter but trapped dust and microbes. And when the neck was broken, microbes could easily enter and one could see microbial growth. So this concept is this experiment is supporting the idea of biogenesis, which suggested that life arises from pre-existing life and not spontaneous generation. Okay. Now let us go through what was spontaneous generation and how things were disproved. So the theory of spontaneous generation was given by the Greek philosopher Aristotle. Aristotle proposed that life arose from, we will check once if anyone is here. Okay. Um, so Aristotle proposed that life arose from non-living material if the material contained pneuma or vital heat. So as evidence, he noted several instances of animals appearing from environments previously devoid of any such animals, such as the sudden appearance of fish in a new puddle of water. So this theory actually had persisted till the 17th century, where scientists undertook additional experimentation to either support it or to disprove it. So proponents of the theory cited how frogs simply seem to appear along the muddy banks of the Nile River in Egypt during annual flooding. Others observed that mice simply appeared among grain stored in barns with thatched roofs. When the roof leaked and the grain molded, mice appeared. So then there was this person, Jan Baptista van Helmont, 17th century Flemish scientist, uh, who proposed that mice could arise from rags and wheat kernels left in an open container for 3 weeks. So actually, in reality, such habitats provide ideal food sources and shelter for mouse populations to flourish. However, they initially thought that mice were arising from rags and wheat kernels left in an open container. Okay. And then the spontaneous efforts efforts were taken to disprove this theory. Uh, Debate continued well into the 19th century, with scientists serving as proponents of both sides, as in either supporting this theory of spontaneous generation or trying to disprove it. So there was this Paris Academy of Sciences, which had offered a prize for solving this problem, because there was a dichotomy here. So then came Louis Pasteur, who eventually did the Swan neck flask experiment, where uh, he filtered air through a gun cotton filter, and upon microscopic examination of the cotton, he found it to be full of microbes, suggesting that exposure of broth to air was not really introducing a life force to the broth, but rather airborne microbes. So he made a series of flasks with long twisted necks. That's where the Tom Swan neck flasks came in which he actually boiled the broth to sterilize it and then allowed air inside the flasks to be exchanged with air from the outside. Like it was open, the mouth was open, but he prevented the introduction of any airborne microbes. So, correctly, he predicted that sterilized broth in his swan neck flasks would remain sterilized as long as the swan necks remained intact. However, should the necks be broken, microbes would be introduced, contaminating the flasks and allowing microbial growth within the broth. So Pasteur's set of experiments irrefutably disproved the theory of spontaneous generation and earned him the prestigious prize from the Paris Academy of Sciences in so long back, which is 1862. Now, in a subsequent lecture in 1864, Pasteur articulated this term omni vivum ex vivo, that life only comes from life. And in this lecture, he recounted his famous swan neck flask experiment, stating that life is a germ and a germ is life, and never will the doctrine of spontaneous generation recover from the mortal blow of this simple experiment. So this is the summary, and this is how the swan neck flasks looked like. And what he also did that he broke the swan neck flask and saw the microbial growth inside upon when this neck was broken. So the curve of the flask actually prevented outside air from entering the flask. This is Louis Pasteur, and there no contamination occurred. And when the neck of the flask is broken off, bacteria reach the sterile broth and organism growth occurs. Okay. Next question is based on the chemical theory of evolution. The Oparin and Halden hypothesis proposes that, and theory of abiogenesis. So the Oparin-Halden hypothesis proposes that life originated through chemical evolution on early Earth. The reasoning statement is early Earth had a reducing atmosphere containing gases like methane, ammonia, hydrogen, and water vapor, which facilitated the formation of organic molecules. So both the statements are true, and the second one correctly explains the first statement. And both these are true, but the reasoning is not the correct explanation of the assertion. A is true, R is false, A is false, R is true. So both these are actually correct statements, like true statements, and the reasoning correctly explains the assertion here. Now let us look at the theory of abiogenesis, which was proposed with the condition that non-living materials can give rise to life in conditions of primitive Earth. The condition of primitive Earth is different from the present conditions, which do not permit abiogenesis. The idea of chemical theory was put forward by these two scientists, A.I. Operan and J.B.S. Haldane. So these were the assumptions. Spontaneous generation of life under the present environment is not possible. Earth's atmosphere is nearly 1 billion years is very different from the current conditions. Primitive Earth's atmosphere was reducing in nature, and under these reducing conditions, the chemical molecules, the inorganic molecules, react with each other through a series of reactions to form organic compounds and other complex biomolecules. And the solar energy and UV radiation provided the energy for chemical reactions. Okay, and this is the Miller and Urey experiment where they tried to, simulate the early Earth conditions. And so definitely the theory of abiogenesis proposed earlier did not find much support without any, strong scientific experimentation. So this chemical theory was experimented by Miller and Urey, where they tried to, simulate or mimic the primitive Earth conditions, where they took a glass flask, then there was a condenser, right? And a liquid flask was interconnected with tubes and a source of electric discharge, I mean, electric spark was there to provide energy. And a mixture of different gases, methane, ammonia, and hydrogen, in a ratio of 2 is to 2 is to 1, along with water vapor, was given at a very high temperature, 800 degrees Celsius. Like all these conditions were trying to mimic the early Earth environment. Okay. So then this was allowed to circulate this mixture in a closed glass apparatus for 18 days continuously, and the energy was provided by electrical discharge, okay, and like in the form of sparks. While passing the mixtures, the gases were passed through a liquid flask to simulate volcano. The mixture was collected from the stopcock and analyzed using chromatographic and calorimetric techniques. Okay. Eventually, upon analysis, it was found that simple amino acids, alanine, aspartic acid, some nitrogenous bases were formed from this experiment. Okay. So, next question is which of the following processes contribute to the primary origin of a new genetic variation in a population? Is it mutation or genetic recombination during meiosis, gene flow or migration, or genetic drift? Correct answer is mutation because mutation is the ultimate source of any new genetic variation. It creates new alleles. Now, when we talk about genetic recombination, it has been asking about, it was asked in the question about the primary source or primary origin. Now, if we talk about recombination, it is actually reshuffling the existing alleles, but it is not creating any new ones. Then if we talk about gene flow, it is actually introducing alleles from another population, but it is not creating a new variation at the species level. When we talk about genetic drift, that also isn't really generating new alleles or introducing new alleles. It is changing the allele frequencies randomly. So mutation is the origin of new alleles, the primary source, whereas all these other options are actually redistributing or altering the existing variations. Okay. Correct option will be only mutation. Now, if you look about just kind of summarize through the things here, um, without genetic variation, some key mechanisms of evolutionary change like natural selection and genetic drift cannot actually operate. So there are three primary sources of new genetic variation, mutations. It changes the information contained in a genetic material. A single mutation can have a large effect, but in many cases, evolutionary change is based on the accumulation of many mutations with small effects. Now, gene flow is actually moving the genetic material from one population to another, for example, through migration, and it is an important source of genetic variation. And then there's mating. It is introducing new gene combinations into a population. And this is genetic shuffling, which is another important source of genetic variation. Next question is, a bacterial culture is subjected to gram staining. After staining with crystal violet and iodine, the cells appear purple. However, after alcohol treatment, the cells lose the purple color and take up the counter stain or saffronin, appearing pink. Okay, so which of the following best explains this observation? So this whole question is kind of, explaining the complete concept of gram staining. So which of the following best explains this observation? The bacteria lack a cell wall, so crystal violet is not retained. The bacteria have a thick peptidoglycan layer that retains the dye complex. Bacteria have a thin peptidoglycan layer and high lipid content, leading to dye loss during alcohol treatment. The iodine fails to form a complex with crystal violet in these bacteria. The correct answer will be the bacteria have a thin peptidoglycan layer with a high lipid content, leading to dye loss during alcohol treatment. So this is gram staining, where you are first applying crystal violet purple dye, and then you are adding a fixing agent, iodine, and then you are washing it with alcohol. Now the ones which are not able to retain the dye loses the purple color and then takes up the counter stain, which is saffronin. So the one which is taking up losing the color and taking up the counter stain, those are gram negative bacteria, and the ones who are retaining the crystal violet dye or purple dye, those are gram positive bacteria. Okay, so this is the overall experiment here. All right. So why is it so? Because gram positive bacteria and gram negative bacteria have different composition of their outer layer, where in gram positive bacteria it's a thick peptidoglycan layer, whereas in gram negative bacteria the peptidoglycan is much thinner, and it is further covered by a stretch of lipids. So those lipids get washed away due to alcohol treatment, and hence the gram negative bacteria loses the very first crystal violet purple colored dye. And if we talk about peptidoglycan here, peptidoglycan is made up of repeating disaccharide unit. One is N-acetyl glucosamine and like or NAG and N-acetyl uramic acid NAM connected by beta 1,4 linkage here. Okay. Next question is, plasmids are run on an agarose gel under identical conditions. The following migration pattern is observed. Band A moves the fastest, band B shows intermediate mobility, and band C moves the slowest, closest to the well. So which of the following correctly matches the DNA forms with bands? So we looked at the plasmid DNA, which can run at different speeds. Right? It can take up different forms and can run at different speeds. So it can be nicked, it can be linear, like both the strands are cut and then it has linearized, the circular plasmid has linearized, or it exists as closed covalently circular DNA. Okay. So the one that which moves the fastest is having the is highly supercoiled or is completely closed. Okay. So the correct option will be the band A moves the fastest because it's completely closed, supercoiled, and overall hydrodynamic radius is the smallest, and it can easily move through the pores of the agarose gel. Then comes the linear DNA, and then comes the nicked DNA. Okay. So here, this is how it looks like. Um, so see, like covalently closed circular DNA or supercoiled DNA is the most compact structure, least friction in the gel matrix. Hence, it is moving the fastest. Then comes the linear DNA, which has intermediate compactness, and it is partially relaxed but still uniform, showing intermediate mobility. And nicked circular DNA is completely relaxed open circle and having an effective, like the largest effective size in gel, showing maximum resistance while moving through the gel, and hence it moves the slowest. So basically, this mobility on an agarose gel is depending on the compactness or how big or small the hydrodynamic radius of the entity is. Okay. Now, a plasmid lacking a selectable marker and origin of replication is introduced into bacterial cells. So, what will happen? Most likely. So, plasmids will replicate independently and be inherited stably. Plasmids will integrate into the host genome immediately. Plasmids will not replicate and will be lost during cell division. Plasmids will express genes but cannot be inherited. So the correct option will be because it is lacking a selectable marker and an origin of replication. So it won't be able to replicate properly and will be lost during cell division in subsequent generations. So without origin of replication on ORI, plasmids cannot initiate replication. So what is happening is there's no replication, so plasmid gets diluted eventually during cell division, and without a selectable marker, even transient presence cannot be maintained in a population. Now, which statement best distinguishes a minimum functional plasmid from a cloning vector plasmid? Functional plasmid requires MCS or multiple cloning sites. Site. Cloning plasmid does not. Functional plasmid requires only ORI. Cloning plasmid requires ORI plus selectable marker plus MCS. Functional plasmid also contain antibiotic resistance gene and promoter. Both types must contain restriction enzyme genes. So, if we talk about a functional plasmid, it requires only origin of replication. Cloning plasmid requires origin of replication, selectable marker to, distinguish between the cells that have taken up this plasmid, with the recombinant plasmid or not. Okay. Plus multiple cloning site. Definitely, this is the site where a gene of interest has to be inserted after digesting with appropriate restriction enzymes. So minimum functional plasmid only needs origin of replication to exist independently. Cloning plasmid needs origin of replication for replication definitely, selectable marker for maintenance in population, and gene insertion site, which we talked about, is the MCS or multiple cloning site. So the minimum molecular components to assemble a bacterial plasmid to perform the function of a vector are as follows. Origin of replication, like any other replicating DNA, plasmids need its own independent origin of replication to provide replication start site to make more copies. It decides the range of bacterial host strain can be used with the particular plasmid vector. The plasmids containing origin of replication region can now be grown in limited bacterial species like E. coli, and in contrast, plasmids containing origin of replication from this particular strain can be able to grow in gram-negative and gram-positive bacteria. So this is just an example, but mainly origin of replication has to be independent, provide a replication start site to make more copies of the plasmid in question. Then you definitely have to have a selection marker in the form of either antibiotic resistance gene or enzymatic gene, which is essential to give phenotypic changes in the host after entry of the plasmid. Promoter, you need to have plasmid replication in the host is performed by the host provided proteins like DNA gyrase, helicase, polymerase, and DNA ligase. But proteins required for conferring antibiotic resistance or enzymes used for selecting transformed host cells is present on the plasmid, and a promoter adjacent is required to express the genes present on plasmid DNA. In addition, promoter is also needed to express the gene present on the foreign DNA. Okay. Now, next, what we have is so during plasmid DNA isolation from E. coli, a student performs alkaline lysis followed by neutralization. After centrifugation, three different types of DNA behave differently. So we have chromosomal DNA, plasmid DNA, and denatured proteins and cell debris. So which of the following best explains why plasmid DNA remains in the supernatant while chromosomal DNA is removed in the pellet? So plasmid DNA is linear and therefore does not renature after neutralization. Plasmid DNA is actually not at all linear. Chromosomal DNA is circular, which is again an incorrect statement and remains soluble while plasmid DNA precipitates. Small covalently closed circular plasmid DNA renatures correctly and remains soluble, while large chromosomal DNA aggregates and precipitates. This is the correct option. Final one is plasmid DNA is degraded by alkaline treatment, leaving only chromosomal DNA in solution. It's the opposite actually. The chromosomal DNA is degraded by alkaline treatment and cannot renature quickly as compared to smaller plasmid DNA, which can renature and form circular DNA again quickly and can move into the solution. Yeah. So this is the correct answer. Okay, the third option. So if we quickly go through the plasmid DNA extraction step, it is a trickier step because plasmid DNA must be kept separate from genomic DNA to avoid cross-contamination. Lysis is more subtle than simply digesting cell wall with enzymes or bashing it with glass beads like mechanical lysis. And so the lysis buffer definitely has sodium hydroxide and SDS detergent, sodium dodecyl sulfate, which can completely denature plasmids and genomic DNA, where it is separating the DNA into single strands. This is actually crucial to perform it quickly because excessive denaturation may result in an irreversibly denatured plasmid. Then we have to neutralize it by potassium acetate solution to renature the plasmid. This is the step where the plasmid and the genomic DNA gets separated. Plasmids being small can easily reanneal forming double-stranded DNA, but genomic DNA is too long to reanneal fully. It tends to tangle so that complementary strands remain separated. And during centrifugation, what happens is any genomic DNA bound to protein forms a pellet while plasmid DNA remains soluble. So at this point of time, one should not be vortexing the mixture or mix this sample vigorously because genomic DNA can break really easily and broken genomic DNA may be small enough to reanneal and go into the solution with the plasmid. Next comes purification. Plasmid DNA is in the supernatant. It can be precipitated with ethanol or cleaned up using phenol chloroform or a spin filter. Spin filter technology neutralization buffer will contain guanidine salts, so the lysate can bind the silica directly for further washing and elution. Resulting DNA is pure enough for most downstream molecular biology applications. The next comes the question on differential centrifugation. A bacterial cell suspension, like fractionation of bacterial cell components. Bacterial cell suspension is subjected to differential centrifugation with increasing centrifugal force in sequential steps. The following components are expected to sediment at different speeds. So if we talk about the ribosomes, nucleoid or chromosomal DNA, cell wall fragments, plasmid DNA and membrane vesicles. So what should be the most appropriate order of sedimentation? So appropriate order of sedimentation will definitely be according to the densities of the um, things involved here, densities of the uh, like molecular entity in question. So ribosomes being the heaviest, most applaudable from lowest to highest centrifugal force required. Lowest one will be so the heaviest one like the are the like ribosomes and membrane vesicles and stuff, but ribosomes are really big in size and dense in size, so it will settle at the bottom or sediment at the bottom. Okay. So ribosomes, then come plasmid DNA, membrane vesicles, cell wall fragments, and nucleoid. So this is what is happening here. Okay. Tissue cells are homogenized and broken or lysed open. Then you have to really need higher centrifugal force. We have to get to the ribosomes. It has to be really high, and subsequent only if we want to get to the cellular debris and stuff and nuclei and stuff, then only this much amount of centrifugal speed is required to harvest those entities. But if we have to harvest the ribosomes, we have to purify the ribosomes, we have to go all the way to really high centrifugal speeds for these ribosomal entities to really, these are dense already, but it is less dense than these things. So it takes some time, like a higher amount of time and higher centrifugal forces to really settle down or to really push them to the bottom of the tube. Now, next question is, during cell division, cytokinesis differs significantly between plant and animal cells due to structural differences. So, which of the following statements correctly explains the mechanism of cytokinesis in plant and animal cells? In animal cells, a cell plate is formed by Golgi vesicles, while in plant cells, a cleavage furrow is formed by actin-myosin contraction. In plant cells, a rigid wall prevents furrow formation, so a cell plate is formed. In animal cells, cytokinesis occurs by cleavage furrow formation. Both plant and animal cells form a cleavage furrow, but plant cells complete it more slowly due to the cell wall. And fourth option is cytokinesis occurs by nuclear division in animal cells and cytoplasmal division in plant cells. The correct option is, in plant cells, a rigid wall prevents furrow formation. So a cell plate is formed, whereas in animal cells, cytokinesis occurs by cleavage furrow formation. Now, if we look at it, so cytokinesis is basically the division of cytoplasm into two halves, indicating the end of cell division process. It occurs immediately after the mitotic phase to separate the nucleus, cell membrane, and the rest of the cytoplasm into two halves to form two distinct and complete cells. The phase begins with the constriction of the cell membrane, which ultimately leads to cleavage and division. Constriction is first observed during anaphase, which continues to grow deeper to finally cause cleavage. The process and mechanism of cytokinesis might be different in different cells. For example, cytokinesis in some cases is often considered to be a part of the mitotic phase, but in the case of animal cells, cytokinesis and mitosis might occur independently. The contraction of the cell membrane during cytokinesis is brought about by contraction of actin fibers that form a bundle called a contractile ring. And in case of plants, a distinct cell plate is formed at the middle of the dividing cell, which separates the cytoplasm and cell organelles into equal halves. And so cytokinesis is also regulated by several factors that are responsible for initiation of division as well as termination. So in case of animal cells, this is the cleavage furrow. uh, these ones here, these indentations here, and this is a parent cell with a contractile ring in between. So contractile ring is formed of actin filaments, forms at the cell center here, and the filaments constrict, forming a cleavage furrow. So from the periphery, the thing is moving towards the center, and eventually the cell is dividing. Okay. However, in case of plant cells, you have Golgi vesicles, which the vesicles accumulate at the metaphase plate here, and the vesicles fuse to form the cell plate, this thick one here, and then eventually the cell plate fuses with the cell wall, forming two daughter cells. Okay. Next, we have the question, during nucleic acid purification, a cell lysate is treated with phenol chloroform mixture followed by centrifugation. Three distinct layers are obtained: upper aqueous phase, interphase, and lower organic phase. Which of the following correctly explains the distribution of biomolecules after centrifugation? DNA partitions into the organic phase, RNA remains in the interphase, and proteins remain in the aqueous phase. Proteins move to the organic phase, DNA and RNA remain in the aqueous phase. DNA and RNA are degraded at the interphase, which is wrong, while proteins remain soluble in the aqueous phase, which is again completely wrong. DNA partitions into the interphase, RNA into the organic phase, and proteins remain evenly distributed, which is also again wrong. So the correct answer is proteins actually move to the organic phase, option 2, and DNA and RNA remain in the aqueous phase. So when a cell lysate is mixed with phenol, chloroform and centrifuged, aqueous phase or the top phase or the clear phase contains hydrophilic molecules like nucleic acids due to their charged phosphate backbone. Organic phase or the bottom heavier phase contains phenol and chloroform, dissolves hydrophobic and denatured proteins. Proteins lose structure and migrate here. When you have interphase or middle layer contains denatured proteins, lipids and debris. It acts as a boundary zone. So in case of phenol chloroform, mixture of buffer saturated phenol and chloroform, usually close to 1:1 for DNA purification with other ratios, sometimes is used for RNA purification. Isoamyl alcohol can also be added as anti-foaming agent. And density of the buffer saturated phenol is little higher than water. So if aqueous phase contains enough salt or any other solutes to increase its density, phase inversion during extraction can happen where aqueous phase goes under the phenol phase. Chloroform significantly is denser than water, so adding into the organic phase increases the overall density of that phase, preventing phase inversion. Chloroform also helps to reduce the interphase, the fuzzy border between the two phases populated by molecules that can't decide where they want to go. And fuzzy border can be due to partially denatured proteins, DNA depending on the pH, or partially denatured DNA binding proteins that are still clinging to DNA. Pipetting off some of this material while removing aqueous phase decreases purity of sample. And also less volume pipetted gives you less yield. So you have to uh, you know, make a balance between how much amount is getting pipetted and we should not be too greedy with pipetting the whole aqueous phase and in the and meanwhile contaminating the tip or the aqueous phase with the interface entities. Okay. Next comes the citric acid cycle or the TCA cycle or the Krebs cycle is often described as a metabolic hub because it integrates multiple biochemical pathways. So which of the following statements best supports the idea where Krebs cycle acts as a master regulator of all metabolic cycles? It directly produces glucose through gluconeogenesis in all organisms. It generates intermediates that serve as precursors for amino acids, fatty acids, or heme synthesis. It functions only in anaerobic organisms to generate ATP efficiently. It is completely independent of other metabolic pathways like glycolysis and beta oxidation. Correct answer is it generates intermediates that serve as precursors for amino acids, fatty acids, and heme synthesis. So Krebs cycle or TCA cycle it is not just an energy yielding pathway, it supplies the key intermediates for biosynthesis like this alpha ketoglutarate which can give rise to glutamate amino acid and other amino acids from there. Oxaloacetate is an intermediate product in the pathway which can give rise to aspartate and nucleotides and amino acids. Succinyl coenzyme A is also part of the Krebs cycle, which is required during heme or porphyrin synthesis. Then there's citric acid, which is a fatty acid and which is involved in fatty acid and cholesterol synthesis. Okay. So if you look at the Krebs cycle here, it is centrally connected to the metabolic intermediates of carbohydrate, protein, and lipid metabolism. It has several branching points where it can communicate with either protein or lipid metabolism. Lipid metabolism is also connected to Krebs cycle through a common intermediate like citric acid and acetyl coenzyme. This one here and this one here. Okay. Protein metabolism also shares intermediate at alpha ketoglutarate here. Here, this one alpha ketoglutarate or oxaloacetate here, this one here. As a result, Krebs cycle can allosterically or through product inhibition regulate other metabolic pathways. In addition, it can redistribute the intermediates between metabolic pathways and hence help in conversion of sugar to protein, lipid, or vice versa. Next, we have the question of Ramachandran plot. All right. Okay. So the Ramachandran plot shows the allowed phi and psi angle distributions for amino acids in proteins. So which of the following statements is the most correct one regarding the behavior of glycine and proline in the Ramachandran plot? So glycine has a higher restricted plot due to steric hindrance, while proline has the widest allowed conformational space. Proline shows very restricted phi angles due to its cyclic structure, while glycine shows the widest allowed conformational space. Both glycine and proline show identical allowed regions because they share similar side chains. This is definitely a very wrong statement. Glycine is the simplest amino acids with two H atoms present, and proline has a circular cyclic ring in presence of like the amino group, and the additional functional group is not free. Glycine is absent from alpha helices and beta sheets due to complete lack of steric effects. So the correct option is like option 2, where proline is showing very restricted phi angles due to its cyclic structure, while glycine shows the widest allowed conformational space. So if you look at the Ramachandran plot, this is the phi and psi angles here. Uh, this one, C and C alpha is the phi angle here, and psi angle is C alpha C bond here, where the rotation is possible, and NC alpha is the psi angle. Okay, and these are the regions where different kinds of secondary structures are allowed. Okay. Next question is, a protein mixture is analyzed using SDS page under different buffer pH conditions. Normally, SDS denatures proteins and imparts a uniform negative charge, allowing separation mainly based on the molecular weight. In an experiment, the following observations were made. When at pH 8.3, the standard condition, sharp, well-resolved bands were observed. At acidic pH 3.5, diffused bands were observed with altered mobility. And at an alkaline pH of 10.5, slightly shifted but resolved bands were observed. So, which of the following is the most appropriate explanation of these observations? SDS loses its ability to bind proteins at any pH other than neutral, causing complete loss of separation. Extreme pH conditions can affect protein charge masking efficiency of SDS and protein SDS complex stability, altering migration patterns. Protein separation in SDS page depends only on the native charge, not on SDS binding or buffer conditions. At all pH values, proteins retain native structure due to SDS, so migration differences are due to protein folding. Correct option is, extreme pH conditions can affect protein charge masking efficiency of SDS and protein SDS complex stability, altering migration patterns. So now if we look at SDS, what it basically is, an ion detergent, okay, and it can actually coat the protein sample with uniform negative charge. And if we talk about SDS, it has a discontinuous buffer system and two kinds of gels here at different pH. First is the upper one is the stacking gel, and this is the resolving or running gel. Stacking gel with low concentration of acrylamide, proteins don't separate here. Running gel has a higher acrylamide concentration capable of retarding protein movement. Now stacking gel with pH 6.8, what happens here is glycine. So the running buffer is tris glycine and SDS buffer at pH 8.3. So glycine switches predominantly to the Zwitter ionic or neutrally charged state. Loss of charge causes the glycine ions to move very slowly in the electric field. And there are chloride ions from tris Cl which move much more quickly in electric field and form an ion front, migrating ahead of glycine. And separation of chloride from tris counter ion, which is now moving towards the anode, creates a narrow zone with a steep voltage gradient that pulls glycine ions along behind it, resulting in two narrowly separated fronts of migrating ions and the highly mobile chloride front in the beginning, followed by the slower, mostly neutral glycine front. Now, all the proteins in the sample have an electrophoretic mobility that is intermediate between the extreme of the mobility of glycine and extremely fast chloride ions. So there are two fronts which sweep through the sample well. and proteins eventually get concentrated into the narrow zone between chloride and glycine fronts. So this switch in the pH is due to the we looked at the isoelectric point and zwitterionic forms at different pH conditions. So the glycine amino acid can actually exist in three charged states. Okay, where this is the state where both positive and negative charge exist, which is the zwitterionic form, and the pi is, you know, zero at this point, and there's no net movement of the molecule towards any of the electrodes. Okay, and based on the increase or decrease in, like, increase in pH or decrease in pH and the availability of H plus ions in the surroundings, eventually the amino acid can become only positively charged or negatively charged. Next, we looked at the polymerase chain reaction, which is a very regular and extremely useful laboratory technique, experimental technique, where mostly you are basically amplifying a small DNA copy into exponential number of copies. And initial denaturing leads to melting of double-stranded DNA into single strands here. Lower temperature allows specific annealing primers for these primers to, for the enzyme polymerase to really add in nucleotides from the buffer, like from the surroundings, and add on to this so as to form daughter strands. Then thermostable DNA polymerase elongates the primers, leading to the elongation phase. And after elongation, the target DNA strands are duplicated, so eventually leading to exponential amplification. Okay, allowing the accumulation of the specific PCR product. So a PCR reaction is set up with the following components: template DNA, primers, DNTPs, and tag DNA polymerase. However, so the template is there, primers are there, nucleotide pool is there, and thermostable polymerase is also there. However, there's no amplification even after 35 cycles. Which of the following is the most likely reason? Primers are present, but annealing temperature is too low. Template DNA is double-stranded, preventing primer binding. Mg2 plus are absent from the reaction mixture. Tag polymerase is replaced by DNA ligase. So Mg2 plus might be absent from the reaction mixture because it acts as a cofactor for tag polymerase activity. Because it is saying that even after 35 cycles, no amplification could be observed. It's a, so even if the annealing temperature is low or something, the primer could have properly bind, like could be bound to the template, leading to some amount of extension. So if Mg2 plus is not there, the polymerase won't be functional. It can't really add nucleotides and annealing, primers, denaturation. Even if these conditions are correct, no extension can happen. Okay. In a PCR experiment, multiple non-specific bands are observed on gel electrophoresis, which modification is most appropriate to improve the specificity? Decreasing annealing temperature, increasing annealing temperature, increasing magnesium concentration significantly increase the cycle number beyond 50. Usually this fourth number is not at all, suggested or preferred. So if one increases the annealing temperature, it might be helpful because low annealing temperatures can lead to non-specific primer binding. Increasing temperature increases the stringency, allowing only perfect primer template binding. And excess magnesium or higher cycles can worsen non-specific amplification. Okay. A PCR is designed to amplify a 500 base pair gene fragment. After gel electrophoresis, three bands are observed at 500 base pair, which was supposed to be the gene of interest size, and 700 base pairs and 200 base pairs. Which of the following is the most likely explanation? DNA ladder contamination occurred, primer dimer formation only. Non-specific amplification and primer dimer formation occurred simultaneously. PCR has selectively amplified only GC-rich regions. So non-specific amplification has occurred and primer dimer has formed simultaneously. So 500 base pair is what we actually needed. This is the correct target product. 700 band is a non-specific larger amplicon, and 200 might be a truncated amplification product or a primer dimer, and multiple bands indicate loss of specificity in PCR conditions. Now, the researcher studies gene expression in two systems, a prokaryotic system and a eukaryotic system, E. coli prokaryotes and human cell line system. So this is what the researcher is observing. mRNA processing is absent in system 1, system 2 it is present. Transcription translation coupling in system 1 is present, in the prokaryotic system is present, in eukaryotes it's absent. RNA polymerase types, it's a single in prokaryotic system and multiple types are present in the eukaryotic system. Introns in genes are absent in the prokaryotic system, E. coli in this case, and present in the human system. So which of the following conclusions is most correct from the observation here? In both the systems, transcription occurs in the cytoplasm and uses multiple RNA polymerases? No. In prokaryotes, transcription and translation are spatially and temporally separated? No, because it's coupled. In eukaryotes, pre-mRNA undergoes processing before translation due to the presence of introns and multiple RNA polymerases, which fits well with the observation given here. Prokaryotic transcription requires RNA splicing to generate mature mRNA. There are no introns. It doesn't really need RNA splicing in prokaryotes. Okay. Prokaryotes have a polycistronic gene. So the correct option will be option 3 here. In eukaryotes, pre-mRNA undergoes processing before translation due to the presence of introns and multiple RNA polymerases. So this is the difference between prokaryotic and eukaryotic transcription from the earlier lecture slide only. Next is the genetic code is described as degenerate but unambiguous. Which of the following best explains this statement? A single codon can code for multiple amino acids, but each amino acid is coded by only one codon. Each amino acid is coded by multiple codons, but each codon specifies only one amino acid. Each Each tRNA recognizes multiple amino acids due to wobble pairing. Genetic code varies significantly between different organisms. So correct option is each amino acid is actually encoded by multiple codons, but each codon specifies only one amino acid. So degeneracy is a concept in genetic code where more than one codon can encode the same amino acid. For example, leucine amino acid has 6 codons. Unambiguous, but a codon always codes for only one specific amino acid, and this ensures accuracy in translation despite redundancy. Next is Wobble hypothesis. Which of the following statements is correct with respect to this hypothesis? The first base of the codon is flexible in pairing with the tRNA anticodon. The third base of the codon shows less strict base pairing, allowing one tRNA to recognize multiple codons. Wobble pairing occurs between mRNA and ribosomes directly without tRNA involvement. All three bases of codon show equal strict Watson-Crick pairing rules. So the correct option is the third base of the codon shows less strict base pairing, allowing one tRNA to recognize multiple codons. So what does Wobble hypothesis suggest? That the first two bases of the codons show strict Watson-Crick pairing, whereas the third base is flexible, or the third position is the Wobble position, and hence this also supports redundancy in genetic code and fewer tRNAs than the number of codons present. A newly synthesized polypeptide in a eukaryotic cell is found to be inactive immediately after translation. However, after processing in the Golgi apparatus, it becomes fully active. So which of the following post-translational modifications is most likely responsible for activating the protein? Removal of introns from pre-mRNA, addition of 5' cap and poly A tail, glycosylation and proteolytic cleavage, base pairing between mRNA and tRNA anticodon. This is the post-translational modification, which is glycosylation and proteolytic cleavage. So post-translational modifications occur after translation is done. And in the Golgi apparatus, glycosylation stabilizes proteins and helps in folding or targeting it to the target site. Proteolytic cleavage activates many proteins like some from proenzymes to active enzymes like trypsinogen to trypsin. So options 1 and 2 here are actually post-transcriptional modifications. These are not really post-translational. And option 4 is a post-translational, like it is part of the translation process where there's base pairing between mRNA and tRNA anticodon and finally a peptide bond formation. A protein synthesized in the RER or rough endoplasmic reticulum is destined for secretion outside the cell, and during processing, it is modified in the Golgi apparatus before being packaged into vesicles. So, which of the following statements is most appropriate regarding this process? The protein bypasses the ER and is directly translated in the Golgi. Signal peptide remains intact in mature secreted proteins. Post-translational modifications in the Golgi determine protein sorting and final destination. Secretory proteins do not undergo any modifications after translation. The correct option is the post-translational modifications in the Golgi determine protein sorting and final destination. Okay, so what happens here is secretory proteins follow this pathway from ER to Golgi and then packaged into vesicles and then secreted out to the target site. So in Golgi, these modifications can occur post-translational ones, glycosylation, sulfurylation, phosphorylation, where proteins are sorted and packaged into correct vesicles. Signal peptides are actually cleaved in the ER, not retained, and protein synthesis never occurs in Golgi. So these are some of the post-translational modifications, okay, that can happen after a peptide is formed or after a protein is formed. Next option is a researcher injects an antigen into a mouse and later isolates antibodies produced by different B cell clones. These antibodies bind to multiple distinct epitopes of the same antigen. So which of the following best describes these antibodies? So these are monoclonal because they are highly specific to a single epitope, or polyclonal because they are produced by a single B cell clone. They are polyclonal because they are produced by multiple B cell clones recognizing different epitopes, or they are monoclonal because they are recognizing multiple epitopes of the same antigen. Correct option is this one. They are actually polyclonal, recognizing different epitopes because these are produced by multiple B cell clones. So polyclonal antibodies are a mixture of antibodies from different B cell clones. Each clone recognizes a different epitope on the same antigen. This results in broader binding but lower specificity as compared to monoclonal antibodies. So this is the same one, same slide taken from the immune system part earlier, where types of antibodies can be polyclonal, which is cheap and quick to produce, high sensitivity, good for detecting low abundance proteins and native proteins, can bind to multiple epitopes on the same antigen. Then there's monoclonal antibodies formed by hybridoma technology, where antigens are injected into the mouse, spleen B cells are isolated, fused with myeloma cells to give it, the stem cell like property for continuous division, and then this forms the hybridoma to produce identical antibodies. It has very high specificity, low cross reactivity. It's very expensive and slower to produce. Okay. So this is hybridoma technology to produce monoclonal antibodies by fusing antibody producing B cells with immortal myeloma cell lines, enabling continuous specific antibody production. Okay, this is from the earlier this thing only. So selection is based on hybridomas cultured in HAT medium, hypoxanthine, aminopterin, and thymidine, which selectively allows only the fused cells to survive. Unfused B cells die naturally, and myeloma cells cannot survive in this HAT medium due to the lack of HGPRT enzyme. Okay, so this is the schematic representation of the same. So what is happening here is um, nucleic acid synthesis is essential definitely for growth and multiplication. So HAT medium inhibits these de novo pathways for simple sugars to form nucleotides. Okay, so de novo pathway is blocked in all the cells. HAT in if like HAT medium is there, and cells with salvage pathways containing the HGPRT enzyme can only survive. So myeloma cells depend solely on the de novo pathways for nucleic acid synthesis, whereas kidney cells has both de novo and salvage pathways. In the presence of HAT, individual myeloma cells or kidney cells will not survive, whereas only hybrid cells can be able to survive. Okay, so that's all. And the questions which about the nervous system and etc. Those are like pretty recent tutorials, and we have looked at many of the question reasoning based questions and stuff. Although I think in NPTEL examination, those are objective type questions only uh, s- with some analytical and straightforward answers both. Okay, so that's all for today's tutorial, and this ends the basics of biology course, and all the best for your exams. I will stop presenting. Okay, all the best for your exams. Thank you.